Please appreciate this presentation has been prepared with all sincerity and to the best of knowledge of students, teachers, and volunteers. We welcome words of encouragement and also suggestions for improvement. For use in academics or any kind of exams, please verify and correlate with your course books. For becoming an associate and or sending contributions in different subjects including storytelling, please get in touch with your brief profile at baskinjoy@gmail.com. So welcome back everyone. Today will be the last lecture, lesson number 4 on the topic tissues in class 9 biology. So first off, we'll be looking at the different types of epithelial tissues present in animals. If you don't know what are epithelial tissues, all the types of animal tissues, I highly recommend that you go and watch my previous lecture in which I give a brief overview about all these types of tissues. So firstly, we have simple squamous epithelial tissue, stratified squamous, columnar, ciliated columnar, cuboidal, and glandular. Now don't get scared by so many types because they're actually really easy. So the first type is simple squamous. They are extremely thin and flat and they form a delicate lining. So in this image you can see that they're very thin and they're flat. So where they're found in the esophagus, the food pipe, and in the lining of the mouth, the inside lining of the mouth. Now next up we have stratified squamous, and you can think by its name only. Stratified means condensed together. So you can see in this picture we have lots of squamous epithelial cells, but they're all squished together. All of them are squashed together. They're condensed. So they consist of layers of simple squamous epithelial tissue to form a stratified layer, and this helps in prevention of wear and tear. So the perfect example for stratified squamous epithelium would be the outer skin of our body. Next up we have columnar epithelium tissue. Now you can probably guess by looking at the picture also, they're in the form of columns, they're upright columns, and these types of epithelial tissues are present where absorption and secretion in the body occur, and they facilitate movement across the epithelial barrier. A perfect example is the lining of the intestine. Now in the intestine, like all the food is digested, all the carbohydrates, proteins, fats, etc. Now that's with the help of columnar epithelial tissues. These help in absorption of material. That's where these are found. Next up we have ciliated columnar epithelial tissues. Now you might know what ciliated means in biology. Ciliated means hair-like structures. So as you can see, these are all individual columnar epithelial tissues. However, on the top you can see these hair-like structures. Now this means this is what ciliated columnar tissue is, and they're present in the respiratory tract mainly, in which these cilia help to push forward the mucus present in our nostrils, for example. Lastly, we have cuboidal epithelial tissue. Now these are there to provide mechanical support. Now they're in the form of a cube. You can guess that by the name cuboidal. And they're present in the lining of the kidney tubules and the ducts of the salivary glands. And they're mainly there for mechanical support. The last one is glandular epithelium. Now you might know, like we sweat, we have oil glands. Now those are all of them are present at the surface. So now this is an example of the basic structure of glandular epithelium. Like like the oil or the sweat is secreted and it gets secreted out on the epithelial surface. Now that's the last type of epithelium tissue, glandular epithelium. Next up, we look at connective tissues and what are connective tissues. Now, cells of connective tissues are loosely spaced and are embedded in an intercellular matrix. Now, this is really important as this matrix can be of many types. It can be jelly-like, it can be fluid, dense, or rigid. Now, what's a matrix? Like as you can see, all of these are individual cells present over here. The matrix basically fills all the intercellular spaces. So the types of connective tissues, we have blood is a connective tissue, bone, ligaments and tendons, cartilage and areolar and adipose. So in blood, the matrix is liquid and we call it plasma. And this matrix contains proteins, salts, and hormones. Now, blood transports all the various necessities like food, gases, hormones, etc. all across the body. So blood has a liquid matrix which is known as plasma. That's the main thing you should know. Bones have a hard matrix and it's composed of calcium and phosphorus compounds. Bones are strong and non-flexible, and because their matrix is basically strong and non-flexible, and bones form the framework that supports the body. Now, many people get confused between these three things: ligaments, tendons, and cartilage. Ligaments join bones and they're very elastic. Tendons join muscles to bones. They are very strong, however, they have limited elasticity. Cartilage are present at the outer surface of bones and it smoothens the edges of the bones so as to provide ease of movement. So this picture shows ligaments, like the ligaments are joining these two bones. They're present in white. Ligaments join bones and they're very elastic, like you can move your bone, you can move your arm, your elbow, etc. Tendons over here, as seen here, they join the muscle to the bone. That's what a tendon does. It joins the muscle to the bone, and they have limited elasticity. This is really important. Like you might have pulled the muscle before. That's because the tendons get stretched to their extreme. Now they need to relax. The tendons have limited elasticity. They can't just stretch forever like the ligaments. The last one, cartilage, you can see like this is the kneecap. This cartilage is present to smoothen the bone surfaces so that there's no creaking noise, so that there's no rough edges. That makes the edges smooth for ease of movement. Now that's cartilage. Lastly, we have areolar and adipose. Areolar fills the space inside organs and supports internal organs, and it by helping in repairing of tissues. So areolar tissue basically repairs the tissues of our body, and it's found between the skin and muscles, around blood vessels and nerves, and inside the bone marrow. Adipose tissue is the fat storing tissue in the human body. That's where all your fat is stored. It's in the adipose tissue and it's found in between internal organs. Next up, we'll have the types of muscular tissues. So the first one is known as striated, also known as voluntary or skeletal muscles. Why are they called striated? Now you might see that we have a dark band over here and a light band. We have dark and light. We have dark, light, dark, light. These are known as striations. These are known as voluntary muscles because they only move when we tell them to move, like they're under our control. And they're also known as skeletal muscles because they're attached to bones. The second type of muscles are smooth muscles, also known as involuntary muscles. Why are they called smooth? Well, that's because they don't have any striations. Like over here, we had black, white, black, white. These are smooth. They don't have any striations. And they're involuntary muscles. Like they're found inside the stomach, inside the small intestine, and they move on their own. We don't have to think about them. Lastly, we have cardiac muscles. They're really special. They have striations, as you can see, dark, white, and they're also branched. None of these were branched before, but these are they're branched like the branches of a tree basically. These are cardiac muscles and they're present inside the heart. So how do muscles really contract? Now muscles contract and relax with rhythmic movements with the help of two proteins which are myosin and actin. The myosin is attracted to the actin. However, initially there are some bodyguard proteins which don't allow this bonding. You'll get to know this later. The release of calcium into the muscle fibers unbinds these bodyguards, and the myosin and actin are able to perform their function. Now, what do I mean? Let me just draw a quick sketch over here. So we have now this over here is our myosin, you can say. It's basically like that. This is just a rough diagram, and on the top we have, this is inside the muscle fiber. So this is myosin and this over here is actin. Now this myosin really gets attracted. It really wants to bind with this actin. However, as I said, there are bodyguards present. Like there's this layer which is present and it doesn't allow this binding. You can think of this as a bodyguard in a way. Now, what happens is if we tell our muscle to move through our nervous system, which I will talk about later, calcium ions are basically sent inside, are basically distributed inside the muscle fiber. Now these are calcium ions over here. All of these are calcium ions. Now these bodyguards, as I said, like these, there are some bodyguard proteins which are guarding the actin. The calcium ions attract these and the bodyguard proteins lift up. So basically now there are no more bodyguards protecting the actin, and this myosin will get attached to the actin. So what will basically happen is the myosin will use ATP. ATP is the energy currency of the cell. It will use this energy and it will fling upwards and get attached to this actin, and it can move the actin in either way. It can go to the right, to the left, or to the right, thus uh, stretching or relaxing the muscle. Now once it has used the ATP, it goes back to its original state, and after some time the concentration of calcium ions embedded inside the muscle fiber is reduced and the bodyguard layer gets back on the actin. So that's basically how our muscles contract and relax. Now this happens in a split second. Now it's really amazing to see that so much activity is happening in a split second. We have the calcium ions lifting up the bodyguard layer. The myosin uses the ATP and it moves the actin layer left or right, thus moving the muscle. And then the calcium ions move back, the bodyguard layer gets attached, and the myosin can no longer move the actin. So how are nerve impulses transmitted? Initially, the axon of the neuron is negatively charged on the inside and positively charged on the outside. Now there are special protein gates in a way located on the axon membrane which allows the movement of these ions in and out of the axon. This movement of ions results in the flow of electricity. There's a gap between neurons. There's always a gap between neurons, and this is called as a synapse. The gap between neurons is called the synapse, and two types of signals can be transmitted uh, across the synapse, either electrical or chemical. So what do I mean by that? It's negatively charged on the inside and positively charged on the outside. So this is an axon of a neuron. It's a rod-like structure, and over here we have protein gates in a way you can call them. These are protein gates. Now so initially it's there's positive charges outside the gate and negative charges basically inside the axon. Now what happens is that if an external stimulus acts on this neuron, these protein gates will open. Thus the protein gates will open and the positive charges will get attracted to the negative, and all the positive charges will rush in when the gates are opened. Now this results in the flow of electricity. This movement of charges results in the flow of electricity throughout the axon of a neuron. Now if you don't know how a neuron is structured, this is basically a brief diagram of a neuron in a way. So the long part is known as the axon. These are the nerve endings, and on the top we have something. Now this is the cell body and these are dendrites. These things poking outside like these finger-like projections, these are known as dendrites. So now we know how an electrical current is passed through a single neuron, but we know that multiple neurons are connected with each other. There will be like hundreds of neurons connected to this one neuron. So how is this signal passed on to the others? Now there's two types of ways it can be passed on: electrical and chemical. Electrical is mainly seen only in the heart, and what electrical does, an electrical synapse is when the entire electrical signal is only passed through this tiny gap between neurons. So what this electrical synapses are helpful so as to get simultaneous movements, so as to get rhythmic movements, and you basically don't have any control over electrical synapses because they keep on happening. So if one heart cell is stimulated, it will pass an electrical synapse, it will pass an electrical signal through the electrical synapse to all the cells in the heart. So you don't basically have to think about it, it's always happening and you can't even stop it, because if this electrical signal is passed to another one, it will pass it on to the next neuron through this electrical synapse. Now these are mainly found in the heart, whereas the other ones, the voluntary actions we do over there, chemical synapses are there. And what chemical synapses are, if this is one neuron, it has an electrical signal, going this way. Now before hitting the synapse is basically like a break in neurons. Before hitting this synapse, this electrical signal is converted into a chemical signal. And then this chemical signal, once it enters another neuron, it gets converted back into an electrical signal. So why are chemical synapses better in a way than electrical synapses? Because you can control them. If an electrical signal gets converted to a chemical one, this other neuron has basically, it can think and it can say that, okay, this I don't really have to act. It's not a big deal. It's a small situation. There's no need to transmit the signal. So you can basically control chemical synapses, whereas in electrical synapses, the electricity is always flowing and you can't even stop it once it has been initiated. So what did we learn today? I know it was a lot of information to take in. So we looked at the types of epithelial tissues, the types of connective tissues. The types of epithelial tissues, we have the simple squamous, stratified squamous for the skin, simple ones for the lining of the esophagus, and then we have columnar for absorption and secretion, and we have a ciliated columnar to help to basically push out the mucus in the respiratory tract. We have cuboidal and glandular, cuboidal for mechanical and glandular for basically secreting oil or sweat. Connective tissues, we looked at blood, we looked at bones, tendons, ligaments, cartilage, areolar and adipose tissue. And we looked at types of muscular tissue. We have cardiac tissue, smooth and striated muscular tissues, and how muscles actually contract and relax. How calcium is basically released into the muscle fiber, with the help of which the actin and myosin can bond together and they can move the muscle fiber left or right, contract it and relax it. And then we also looked at how an impulse actually travels throughout a neuron and between neurons. So it's positively charged on the outside of the neuron, negatively charged on the inside. When an external stimulus is provided, the protein gates are opened and the ions can flow in and out. This results in an electrical signal throughout the axon of the neuron, and between two neurons there's a junction, there's a gap known as a synapse. And there are two types of synapses: either the direct electricity can pass through and to the next neuron, and there are also chemical synapses in which that electric current is first turned into chemical signals, which is then later turned to an electrical signal by the second neuron. So that's it for today. I hope you understood at least something. I know it was lots of information, and this will be the end of tissues, and I'll see you next time.